Okay, today we're going to learn a little bit about bacterial genetics. Most simply put, genetics is a study of inheritance, um, and what you're doing when you're studying inheritance is you are looking at the genes of that organism. Um, the genes of a cell's organism, of an organism's, uh, is always going to be what directs all the function that that cell will be able to do. Okay. Um, Another way of thinking about gene is just to say that it's a segment of DNA or of the chromosome uh, that encodes a gene. Now, where bacterial genetics gets kind of different from, uh, for example, Mendelian or human genetics, is that bacteria really don't have a way to have sex. Okay, they have three ways of doing genetic exchange, but it's not male and female sex as we would tend to think about. Um, so the first one of these methods is transformation. And transformation is to take up a free DNA from a donor cell, and then that free DNA can go into the cell. So in transformation, DNA is the only thing that's moved. Now, in transduction, here the DNA is coded by a virus. The viruses would be a bacteriophage, and so that virus is delivering the piece of DNA to the recipient chromosome. And then in the third case, this is a process called conjugation. And in the conjugation process, there's actually a special plasmid or a piece of DNA that's outside of the chromosome that gets moved from one cell to the other cell. Um, and all three of these ways are uh, all sort of equal in terms of being able to exchange genetic material. There's well, there are certain conditions that have to exist for each one of them to occur, and we'll talk about that in a minute. Okay. Now, when we're thinking about genetics in general, um, we're often looking at mutations. And a mutation can quite simply be defined as a change in the DNA sequence. And we can have three categories of mutations: beneficial mutations, harmful mutations, and silent mutations. Um, when a lot of people think about mutations or mutants, you very often go right to the harmful, but that's only a subcategory of them. They're the one that generally is easiest to see. You know, you see a mutant organism and you're like, whoa, what happened to that? But there are beneficial mutations, um, and those beneficial mutations would clearly be what is driving evolution. Um, okay. Um, if we expose uh, the DNA to some physical or chemical agent that makes more mutations, those physical or chemical agents are called mutagens. Okay. Now here's some examples. Starting on the right side, here we have an A going to a G mutation, and the amino acid that they code for is both the same. So even though there's a change in the DNA sequence from an A to a G, since the amino acid sequence is the same, we don't see any difference. Okay. Now here with at the valine with the T going to a C, yes, it's a change in the DNA sequence, but because valine and alanine are very similar amino acids, it's called a neutral mutation. Now, these other two are kind of fun. Here, changing that C to a T gives us a change in the amino acid sequence of proline to leucine. And so this is a missense mutation because that single base pair caused a change in the amino acid sequence. So it's still making something, right? It's still making an amino acid, but it just doesn't make the correct amino acid, so it's a missense. And here finally, the nonsense mutations, here if we take the A and make it into a T, now it doesn't code for any amino acid, so when you don't code for anything, you become a nonsense mutation. Chemical mutagen. And one example of these chemical mutagens is EMS, ethyl methane sulfonate. And there's a structure here on the left. If you know any organic chemistry, you can probably tell that with the sulfur and all these oxygens, this is a very reactive chemical. And so this ethyl methane sulfonate will interact with one of the bases, in this case guanine, the G base. Okay, and what it does is it adds itself to the oxygen here at the O6 position. Okay, and what that does, normally you should be able to base pair to the cytosine at the two nitrogens and the oxygen, but because this group is added to that oxygen, we can no longer bind anything there, and so we make hydrogen bonds, but they're not the right hydrogen bonds, so it inappropriately binds with thymine now instead of cytosine. Now, there's a test that's pretty widely done, and you'll probably see, if you ever read the little inserts on any pharmaceutical you get, any drug you get, it'll say how it did in the Ames assay. And the Ames assay is simply a test to look for if a compound causes mutations. So how you do this experiment is you take a Petri plate and you put down a bunch of salmonella cells, and those salmonella cells, they are lacking the ability to make their own histidine, and so they can't grow on the plate that you put them on. Only if a mutation occurs will you be able to get colonies to appear. Okay, so you put your chemical down, your drug down in the center, and then if nothing occurs, if no mutations happen, then you get no colonies around that little disc where you put your chemical. But if that chemical makes a bunch of mutations, you actually get colonies on this plate, and the number of colonies will tell you how strong of a mutagen it is, how many mutations that compound makes. So here are some real plates. In panel A, that's kind of the spontaneous mutation rate. There's not a disc in the center here with the compound, but there are some of these His plus colonies that grew up, so that would be the spontaneous mutation rate. Just by being on the plate, some mutations occurred. B, C, and D, you can see there's kind of a zone around this where we get increased number of mutations, and B, C, and D are all different compounds, and they all induce different amounts of mutations, so that, well, we see the most colonies in D, so that would probably be the strongest mutagen. It's making the most mutations, because that's where we get the highest number of mutants. Bruce Ames did an experiment, and he actually developed a series of those Salmonella strains where he can put known amounts of the mutagen onto the plate, and then count how many colonies arise from it. And so here in the strain TA100, we get a great increase in the number of mutations. In TA1538, we get very few mutations. And Bruce did a really nice thing, and that is that he can tell the specific mutation that it took to become histidine plus by which strain he's in. So he can know, so here we're looking at the compound of aflatoxin B1, that B1 makes a certain mutation, which is a G to A mutation, and we actually know something about the specificity of the reaction now, because the aflatoxin is only increasing strain TA100, and it's not increasing in the 1538 or 1535 strains. Okay. Just a little side note on this aflatoxin B1. Aflatoxin B1 is a toxin that is made by fungus, and it's actually a very, very common fungus whenever you store grain. And in parts of the world where grains have to be stored for a very long time, you get a very high increase in aflatoxin B1. And you get more human cancers because the grains have that aflatoxin B1 in it.